RNA primer is required for the synthesis of a complementary strand during replication. So, the requirement of RNA primer to initiate all new DNA synthesis creates a dilemma for the replication of the ends of linear chromosomes. And this problem is called as the end replication problem, and this only occurs in case of linear molecules or linear chromosomes. This difficulty is not observed during the replication or duplication of the leading strand template. As you know, along the leading strand, when the replication takes place, there are two strands. One is lagging strand, one is leading strand. So for leading strand, only one RNA primer is required, and one RNA primer attaches to that strand, and it initiates the synthesis of the complementary strand, and this complementary strand synthesis completes up to the extreme terminus of the chromosome. So only one RNA primer is required, which completes the synthesis along the leading strand. In contrast, the requirement for multiple primers to complete lagging strand synthesis means that a complete copy of its template cannot be made. Along the lagging strand, the synthesis takes place in the form of fragments, and these fragments are called Okazaki fragments. And for each Okazaki fragment, there is separate or new RNA primer. So a number of RNA primers are required. So that's why, due to that requirement, the synthesis of a new strand along the lagging strand that cannot extend up to the extreme of chromosome. Even if the end of the last RNA primer for Okazaki fragment synthesis anneals to the final base of the lagging strand template. For example, these are two strands, one is leading, one is lagging. So we suppose that for the synthesis of last Okazaki fragment, the RNA primer attaches to the extreme end of this strand. Even then, it is not possible, because once this RNA molecule is removed, there will remain a short region. This region will remain, which is unreplicated, single-stranded at the end of chromosome. Here in this picture, you can see this is a linear chromosome or molecule of DNA. These are the replication forks or replication bubbles, you can say. And at the end, when the replication completes, here you can see this strand. This blue one is complete, while the yellow one is incomplete at the terminus or at the telomere. Similarly, on the other molecule in this, again this green one, which is leading strand, this is complete, and the other one which is yellow, this is incomplete. So in case of linear chromosomes, always at the completion of replication there will remain some unreplicated ends. Telomerase, this is another important enzyme used during replication, is a remarkable enzyme that includes multiple protein subunits and an RNA component. So this is a multimeric enzyme. It contains many protein subunits and especially RNA component as well. Like all other DNA polymerases, telomerase acts to extend the 3' end of its DNA substrate, like all the polymerases do. But unlike most DNA polymerases, telomerase doesn't need an exogenous DNA template to direct the addition of new dNTPs. This is a different feature from other polymerases. Instead, the RNA component of the telomerase serves as a template for adding the telomeric sequence to the 3' terminus at the end of the chromosome. Because you know this telomerase has its own RNA component. So that RNA components serve as the template. Telomerase specifically elongates the 3' OH of the telomeric single-stranded DNA sequences using its own RNA as a template. So what telomerases do? It elongates the 3' OH of the single-stranded or lagging strand using its own RNA as a template. So only this strand is extended. Note, new synthesis takes place, only this strand is extended by telomerase. As a result, the new synthesized DNA is single stranded. So as I say, only this strand is extended. So this is new synthesis. So this telomerase results in the synthesis of new single stranded DNA. So it is only extending the extreme end of the DNA strand. So when telomerase acts on the 3' end of the telomere, it extends only one of the two strands of the chromosome. Then how the 5' end extends the other, which was already short? This was actually completed, and this strand, the 5' end, this was already short. So how this will be completed? This is accomplished by the lagging strand DNA replication machinery. By providing an extended 3' end, telomerase provides additional template for the lagging strand replication machinery. So this additional sequence, it provides the additional strand for lagging replication machinery to use this additional strand to complete the synthesis of this strand. By synthesizing and extending RNA primers using the telomerase extended 3' as a template, the cell can effectively increase the length of the 5' end of the chromosome as well. So here in this figure you can see this situation is at the end of replication. So this strand is complete, but this one is incomplete. So this is this green one is telomerase. It has its own RNA component, which you can see this yellow sequence. This is RNA component, and by attaching to this end, this extreme end, it starts synthesis of, or it starts extension of this end. So this orange strand, this is a newly extended strand. So after this extension actually will provide a base for this RNA primer to attach, and then this Okazaki fragment will complete the replication. So in this way, both the ends, 5' end and 3' end, will be equal.